Hello everyone, I am Roxana Akhtar Ruma, a doctoral student at Sochi University. My enrolling year is 113, student ID is 1133302120, and my advisor is Professor Dr. Ongkong Sai. This is the progress report reviewed by my professor. This is the timeline of my semester. Here I started my semester 22 February 2025 to present, and I completed my mandatory courses and started my research work. My research work is to check the safety assessment of 4 types of exosomes. This is my coursework. Here I completed total 2 credits. My course name is Sochi Humanity and the Role Model in Biomedical Science and Interdisciplinary Seminar on Medicine. Now I will move to the research work outline. My doctoral research is titled Safety Assessment of 4 Different Sources of Exosomes. This work explores the retinal effects of exosomes derived from 4 different origins when administered into the eyes of long-even rats. This is the retinal toxicity evaluation. Here, each rat received a 5 microliter intravitreal injection of one exosome type. We divided the animals into 5 groups: normal, exo1, exo2, exo3 and exo4, with 5 to 10 rats per group. Across 4 weeks, we monitored retinal changes using optical coherence tomography, electroretinography and flash visual evoked potential. Here we perform OCT profiles at day 28 post exosome IVI to check the retinal thickness and the result shows that none of the exosome types significantly affected retinal thickness in treated eyes as compared to the normal group. This suggested no overstructural damage. Then we perform ERG to check the ERG profile at day 28 post exosome IVI. The result is under dark adapted condition, ERG recordings showed altered A wave and B wave responses in the exo1, exo2 and exo3 groups, while exo4 did not show significant changes. In contrast, under light adapted conditions, all groups preserved photopic function. FVP results confirm no vision loss, indicating that exosome treatment did not impair vision signal transmission. So the summary of the findings are: our primary findings indicated that administration of exo1, exo2, exo3 and exo4 did not significantly alter retinal thickness in the LE rats' eyes. However, ERG data showed that under dark adapted condition, both A wave and B wave responses were altered in exo1, exo2 and exo3 groups, while exo4 showed no significant changes. In contrast, light adapted A wave and B wave responses remained relatively unchanged across all exosome treated groups compared to the normal control group, suggesting preserved photopic retinal function. Additionally, FVEP analysis showed no significant differences between the exosome treated groups and the normal control groups, indicating that no vision impairment occurred following exosome administration. So, the future work will be performed IBA1 and ED1 immunostaining to detect and access activated microglia, conducted GFAP staining to evaluate the astrocyte profile and the potential gliosis, and finally use rhodopsin staining to examine the condition and distribution of photoreceptor cells. Thank you.